In this video, we will discuss about different kind of glycans that are found in the cancerous cells. In the mammalian cells, in general, there are two kind of glycosylation that are commonly observed. First one is the N-linked glycosylation, and second one is the O-linked glycosylation. So in the N-linked glycosylation, the carbohydrate structures are usually formed at the aspargine residue of the polypeptide chain, and there is a common structures in the N-linked glycosylation, that is, it contains two glycan structures and one mannose structures. And one of the common changes that are found in the cancerous cells N-linked that are found in the cancerous cells about the N-glycans is that the aberrant branching or abnormal branching of the N-linked glycosylation, and there are different glycotransferase genes that could be responsible for this extended branching of the N-linked glycans in cancerous cells, and some of them are mentioned here. For example, this MGAT5 or MGAT5B helps in the helps in the beta 1,6 linkage to this mannose structure of this glycan, whereas MGAT3 helps in the glycan this beta 1,4 linkage of this glycan to this first mannose, and MGAT4 helps in the beta 1,4 linkage of this glycan to the second mannose. That can be seen here. So this is how the abnormal branching is formed, one of the way by which abnormal branching is formed in the cancerous cells. And in the case of fucose cells, in the case of fucosylation, not fucose cell, in the case of fucosylation, what happens is that in the N-linked glycosylation, the FUT8 helps the fucose addition to this first glycan structures via alpha 1,6 linkage, whereas in the case of O-linked glycans, different fucosylation glycotransferase enzymes plays different role. For example, this FUT3 helps in the alpha 1,3 or alpha 1,4 linkage of this fucose to this glycan structures, whereas FUT4 to 7 and 9 to 11 helps in alpha 1,3 linkage of this glycan to this fucose monosaccharide, and whereas FUT1 or FUT2 helps in alpha 1,2 linkage of this galactose to this fucose. So this is how the different fucosylation structures are formed in the cancerous cells. And after this, or apart from this aberrant glycan structures, there are other glycan structures are also found, and one of them is mentioned here, that is the galactose addition to the GalNAc can be done with the help of enzyme B4-GalNT2, and this helps in beta 1,4 linkage of galactose with this GalNAc, and these are also found in the case of cancerous cells. And now the important things that are more predominantly exist in the cancerous cells is the silation. So different silations has been found in the different cancerous cells, and one of the reason could be this kind of structures could be formed because of the upregulation of ST6-GAL1 that helps in alpha 2,6 linkage of galactose to the sialic acid, and after that the ST8-CR1 helps in alpha 2,8 linkage of the first sialic acid to the second sialic acid. And in the case of second structures, this ST3-GAL1 to 6 helps in alpha 2,3 linkage of galactose to this sialic acid. So these are also found in the case of cancerous cells apart from the other mentioned here. And lastly, we will discuss about what are the glycotransferases that are responsible for the truncation of O-glycan in the cancerous cells. And this one is the most common, that is TN and STN antigens are predominantly found in the pancreatic cancers. So this could be because of this STN region could be because of the upregulation of ST6-GalNAc1 or ST6-GalNAc2 that helps in the alpha 2,6 linkage of GalNAc to this sialic acid, whereas upregulation of GalNT2, 3, 5, 6, 7, 10 helps in the bonding of this serine thiorin at the polypeptide chain to this GalNAc monosaccharide, and thereby producing more truncated O-glycan structures. And other truncated structures can be formed at core 1, core 2 and core 3 with the help of this enzyme C1 GALT1 beta 1,3 that helps in this structure formation, that is GalNAc will bind to this galactose. This is the core 1 structures, and the upregulation of GC and T1 helps in beta 1,6 linkage. GC and T1 helps in the beta 1,6 linkage of GlycNAc to this galactose, and this is and after that the other glycotransferase genes that is responsible for further extension of this O glycans is this B4 gal and B4 GALT 1 to 3 and 5 that helps in beta 1,4 linkage of GlycNAc to this galactose. And then lastly or finally the core 3 glycan structures can be formed with the upregulation of beta 3 G and T6 that helps in beta 1,3 linkage of GalNAc to this GlycNAc. So these are some of the common structures that are found in the cancerous cells, and what could be the possible glycosyltransferase changes that could result in such kind of glycan structures. If you need to understand more about this different kind of glycans in cancerous cells, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.